We're going to take notes now on Unit 1, Lesson 12. This is 1.12, so please make sure that if you need to, pause the video to get a new note set up so that you're ready to go. Alrighty, let's proceed. So the next two terms, and actually the final two terms that we're going to discuss in this unit are exo and endocytosis. And you can see actually over here in my diagram a visual representation of what these two things look like. So if you are confused as to what I'm referring to, maybe listen to me and look at the diagrams underneath exocytosis and endocytosis to get a visual as to what I'm referring to. So the first term that we're going to start with is exocytosis. I'm sorry, endocytosis, my bad. Endocytosis is where we have cells that move particles across the cell membrane and into the cell. So in endocytosis, this is going to be the particles that are going to come into the cell membrane are going to be too large to just easily pass through the cell membrane like gases such as like carbon dioxide and oxygen. So these are going to be larger particles that need to be brought into the cell. An example would be like the cell membrane surrounding particles of food and bringing it into the cell. And amoebas do this where they will surround their entire bodies around the food particle they want to ingest, and then they will pinch their cell membrane together to create a vesicle around that food particle to then digest it. When you think of endocytosis, think enter. So you can see enter right here is endo. So that was one way to think about things entering into the cell. So this food particle is entering into the cell. It's too large for it just to pass through, but the cell membrane has decided this is a good thing. Remember, our gatekeeper of the cell. So it's going to envelop itself around it. And this is the vesicle I'm referring to, the kind of like little envelope that the food is surrounded in, so then it can then be transported throughout the cell. Now, the other term that we're going to be referring to is exocytosis. And this is where cells will move particles out of the cell and across the cell membrane. So remember, once again, things are too large to just easily pass through the membrane. So the cell is going to surround it in a membrane or a vesicle to get it out of the cell. So, for example, we all are familiar with this, that Golgi apparatus that's going to package up the proteins that are sent to it that are made by the ribosomes. The Golgi apparatus, they will do exocytosis to get the proteins to go out of the cell. So, just like endo, think in or enter, with exo, think exit. So, they both start with X. So, this is the same type of thing. Once again, you have maybe these are the proteins that were made in the Golgi complex. It's too big to pass through, so it's added to the cell membrane. The cell membrane will actually dip to attach to the vesicle and release those particles out of the cell. Now remember that since this is going to require energy, both of these processes, exocytosis where things exit the cell and endocytosis where things enter the cell, are going to be active transport because they require ATP. Now, this is a hyperlink to a video that I will attach in the Google Classroom as well that you can watch. It's optional if you'd like to get further explanation on exocytosis and endocytosis. But the tip that I want you to remember is that think enter with endo and think exit with exo.